Assalamualaikum and good afternoon, ladies and gentlemen, and good morning to Bob and Santi in UK. My name is Melinda. On behalf of FOSI committee, it is my pleasure to welcome you here today in FOSI online talk series. I hope you enjoy your lunch already and ready to hear the presentation from Bob. And hope everybody doing well. Stay at home and keep physical distancing. I would like to extend my gratitude to our sponsors of Noble Energy Resources and Geoscience Delta Andalam for funding of this Zoom platform. So now with us together already join the FOSI committee, Mas Ricky, our FOSI General Secretary, and then I see Pak Win also joining us, and the rest will joining us soon. We will also have a Peter Lang register with us and Peter Woodruff. Glad to know you joining us this afternoon. We're glad to inform you that we established the FOSI YouTube channel. We uploaded all the videos from previous talk already. And don't forget to subscribe and like the channel and enjoy the video. Definitely, today's talk will be uploaded as well. So I would like to thank you for joining us today for this special presentation on Palynology in Indonesia, an overview of Quaternary studies, Cenozoic stratigraphy palynology, and sequence biostratigraphy. The registered participants is 250. It's beyond our expectation in this weekend. And get ready to be excited. The presentation will cover the definition of palynology, modern pollen rain and pollen transport, later quaternary studies, palynological zonation for Indonesia, and sequence biostratigraphy. Moving right along, it is now my pleasure to introduce our guest speaker. Really honored to have Bob Morley from Palynova Limited. Many thanks for sparing your time in weekend for us, Bob and Santi. I believe most of you know him very well, met him maybe, or maybe already have his useful publication for Indonesia. I read many publications from Bob. Well, before I hand over to Bob, please allow me to give you some rules in order to make this meeting running smoothly. I would like to get your attention for a moment. First, I recommend all participants to mute the audio and switch off your video during explanation from Bob to maintain good connection. Second, the presentation may be recorded for rescreening within the FOSI platform. Last but not least, a Q&A box can be found in the icons panel, usually at the bottom of your screen. Enter your name for the committee to collect after the presentation. It will be answered live by Bob later on, and you might turn on your audio or video to deliver your question. So now, well, ladies and gentlemen, please enjoy your talk and please welcome Bob Morley. Yes, please, Bob.
Okay, thank you very much. Right, well, I'm Bob Morley. As Melinda explained, I think probably most of you already know who I am. You may be forgetting who I am because I haven't visited Indonesia this year because of the COVID emergency. And so we are isolated here in our house in the UK, and we can only talk to people over Zoom. And well, that's one of the marvelous things about today's technology. We can do that today, but if this happened 10 years ago, we wouldn't have been able to have this webinar at all. Anyway, Melinda already given an overview of what the talk is about. So to save time, I won't repeat that. I just want to emphasize that Palynova is just a small consultancy company, but it's now in its 28th year. We've been doing this now for 20, so soon we'll be 30 years old. And it's just myself and Santi, but we work in Indonesia in association with a company called Exindo Pertama. So if we're working in Indonesia, we work through them, and we have another company that we work with in Malaysia. So with these contacts, it allows us to work across Southeast Asia. Right. Let's. I won't go through this again because Melinda has already explained basically what the program is. So let's go straight into asking what is palynology. well, I'm sure you all know, but just to remind you, the name Palynology actually comes from the Greek word meaning the study of dust, or to strew or sprinkle. And it is in fact the Greek word for pollen, but the Greeks didn't differentiate. They didn't have a different word for pollen than for other dust. And it was introduced in the 1940s because up until that time, the process of palynology was called pollen statistics. It basically started out just looking at pollen and spores. And the good thing about the name pollen statistics, it emphasized that it was a statistical method, that you had to count statistically viable populations of palynomorphs, and then you could analyze them statistically. But the word palynology was brought in because people started finding things that weren't pollen or spores in their samples, and they needed a name that would encompass all of that was appropriate for people working on, say, dinoflagellates or acritarchs or cuticle fragments or palynofacies and things like that. So actually, I think it was a very appropriate name, and it's been used ever since the 1940s. Something to bear in mind is that palynology is an interdisciplinary science, and it really stands at the intersection between earth science and biological science. And it's particularly useful in taxonomic botany and in plant or paleoecology. And basically, as stratigraphic palynologists, we're really looking at the paleoecology through time and trying to date rocks by understanding the former paleoecology and determining whether there were plant taxa around which are maybe now extinct that might allow us to date particular sediments. There's two main divisions in palynology, either paleopalynology, which includes stratigraphic palynology, which is probably what most of us are, and it's a branch of micropaleontology and paleobotany. And this, of course, is a tool for determining the age and the depositional character of sediments and past environments and climates. But the other half of palynology is actuopalynology, and this is a study of pollen and spore morphology, and it's used in plant taxonomy. Now, the point I want to emphasize is that if we didn't have actuopalynologists working on pollen and spore morphology, we wouldn't know what it is we're identifying, and so we couldn't do our job. So these two disciplines really come together, especially in the Cenozoic, where many of our pollen types can be identified with modern plants. So as well as having geological and paleoecological applications, the fossil record from Indonesia helps in dating evolutionary trees based on molecular phylogenies. And actually, this is an ongoing and very active area of palynological research. And you'll see as we go through that we stumble across this from time to time. So palynology in Indonesia, I think Indonesia is a really special place for doing palynology because it's the perfect setting for bringing together paleopalynology and actuopalynology. So we can date the sediments, we can interpret landscapes, determine history of vegetation, biomes and past climates, identify and date plant migrations that relate to plate tectonics, determine the fossil record of pollen types that characterize specific clades that identify that can be identified from molecular analyses, and thus help clarify the pattern of evolution of major plant groups. So it really is a genuinely interdisciplinary science. You know, we find pollen types that will help botanists to understand the process of evolution of groups that they're working on. On the other hand, we find those pollen types and we can use them to interpret stratigraphy and paleoenvironments. So in this discussion, I want to first address how palynology can be used in the late Quaternary. That's the last glaciation and the Holocene. And the purpose of this is to examine vegetational history and climate change since the last glacial period. This is probably where palynology really started in Indonesia, with studies, including my own in Sumatra in the 1970s. I want to look at the early Pleistocene as well, Java man localities, determine the landscape in which Homo erectus was living and the climate, but also we can use sequence biostratigraphy, the method of sequence biostratigraphy, to date Homo erectus fossils very accurately. And this is only just published, this work, but the method developed in the petroleum industry can be used to accurately date the age of Homo erectus. And then we want to look at the methods of the stratigraphic palynologist and look at the succession, the palynological succession from the Paleocene through to the Pliocene, which is of value in geological studies and in the petroleum industry, and probably what most people listening to this are going to be interested in. I also want to mention the implication of the palynological record with respect to plate tectonics. And then finally, I want to discuss sequence biostratigraphy. I think sequence biostratigraphy is the process of identifying depositional sequences from the examination of microfossil distribution patterns. And it mainly involves integrating palynological and foraminiferal data. And it is, in my opinion, the way forward for the stratigraphic palynologist. If you get a palynologist, micropaleontologist that can work really well together, you can produce some really excellent results from that. But first of all, I want to say a little bit about naming fossil pollen because when I see papers published from Indonesia, I always think, there's some issues here with naming. So I just want to run through the different issues and problems about naming palynomorphs in Indonesia. So palynologists working on the Quaternary identify pollen by comparing fossil pollen with modern reference material taken from named herbarium specimens. And my feeling is that all palynological laboratories should have a good collection of modern pollen from their specific area. And you can classify these by family or by pollen type. If you just try and identify your pollen from the literature, you'll never be as accurate as if you can compare it with the actual specimens from modern herbarium material, because you can look at that herbarium, those modern pollen slides using LO analysis, different magnifications, really get a hand on what each particular pollen looks like. And you'll very rarely get all of that detail by looking at a description in a paper. If we look at recent pollen, identifications can be made at different levels of certainty in different taxa. And plant taxa are very general, sort of broadly divided into two groups. You either have Eurypallinus families of plants or Stenopallinus families of plants. And the Eurypallinus have many pollen types and the Stenopallinus have very few. So the best example of a Eurypallinus family, or very good one, is the Lythraceae, that includes the genus Soneratia. And in fact, all the species in the genus Soneratia can be identified on pollen. There's only a few genera in which this can be done. So if you find a tree of Soneratia and you don't know which it is, you can look at the pollen and you can work out which species it is, even if you're not sure about how to describe it from the leaves. So we could talk about three species that are common in Indonesia: Soneratia casularis pollen or Soneratia alba pollen. These are plant type. The pollen actually identifies the plant species. Only a few genera where that happens. Another genus is Nipa. Now Nipa has got very distinctive pollen and it only has one species. So if we find Nipa pollen, Nipa, we know that it's Nipa fruticans because there's only one species within Nipa. But another genus that I've spent a lot of time looking at and published on is Elangium. Nearly all of the species in Elangium can be identified on pollen characters, but there are some that can't. And so often you find species pairs where maybe two species have the same pollen type. And we can indicate this if we find a pollen type that we think is one of those species. And so, for instance, two species that occur in Sumatra, Elangium rotundifolium and Elangium kertzii. If we find one of those, we can call it Elangium kertzii type pollen, and that tells you that it also comes from another species. So it gives you an idea of the precision of our identification relative to modern plants. However, as I said, there's only a few genera where the pollen varies at the species level, and as a rule of thumb, most pollen types can compare to plant genera. And I've given a list here: Durio, Ilex, Onchosperma, Pometia, for instance. If you find these pollen types, you know it's just from one genus. So you can call this pandanus pollen or Pometia pollen, and it's very clear what you're trying to put across. But there are some closely related genera that have the same pollen type. So for instance, if we find oak pollen, it might be from Lithocarpus or Castanopsis. These have the same pollen type. So rather than to say Lithocarpus castanopsis pollen, we simply say Lithocarpus type pollen. And that tells us that there's another genus that has the same pollen type, and we can go back to the literature and work out what that is. Another one is Casuarina. So Casuarina is a tree of that most people know as a tree of coastlines, coastal sand dunes. But Gymnostoma is the tree that produces the same pollen in the same family that is the dominant tree in many peat swamps. And probably most of the pollen that you see that is fossil comes from Gymnostoma, not from Casuarina. So if you find this pollen, you call it Casuarina type, and that will inform you again that it's not exactly from Casuarina. It could also be from Gymnostoma. And we can carry on. You know, several genera here all have exactly the same pollen type. So if we call it, say, Terminalia pollen, Terminalia type pollen, then when we're interpreting it, we could realize, well, it may not really be Terminalia. It could come from Dactylocladus or Melastoma. But if we call it Terminalia type pollen, people will understand that, yeah, it could come from a different genus. And we can continue. If we look at the Stenopalinus families, look at grasses, then all grasses produce the same pollen type, and we can call it Poaceae pollen. This is a present name for the grass pollen family. But these two families, which you'll find pollen of quite regularly, Chenopodiaceae, Amaranthaceae, these have the same pollen type. And Americans call this Chenoams, but that's a very colloquial term. If you call this Chenopodiaceae type pollen, it will tell you that this pollen could come from either Chenopodiaceae or Amaranthaceae. Now, this approach was suggested by Anderson and Muller in their 1975 peat swamp paper. It's very simple and unambiguous, and I think it would be very useful if it was followed more widely. Now, a lot of people, when they're palynologists, when they're naming fossil pollen, especially from the stratigraphic palynology perspective, add SPP after their name. So here we're dealing with pollen types that relate to clades, not to species. And so, although you can use such a nomenclature for a foraminifera, you shouldn't use it for pollen. The only exception is if you're looking at genera like Sonratia and Elangium and you can't work out which species it is. Otherwise, you should just call it, name it after the genus or the genus group, not add SPP because it's not relevant. And this becomes more apparent when we get into the Cenozoic. And for the Cenozoic, your preferred approach, or at least my preferred approach for naming dispersed fossil pollen, is to use dispersed fossil nomenclature where appropriate names have been published. Now, only a few names so far have been published. There are many types that have not been published from Indonesia or Malaysia. And so if I just go through some of those pollen types that we discussed before, and look at their the equivalent nomenclature using dispersed fossil pollen names, then you can see the issues. For instance, if we find Soneratia alba pollen, we'd name that Florschuttia meridionalis if we found this, say, in the Miocene. And if we found Florschuttia trilobata, we could legitimately call this Soneratia sp, because it's from an extinct species of Soneratia, and we don't know what that species actually was. If we find Lanagiopolis nangulanensis, then this comes from either Elangium ebenaceum or a different one other species has the same pollen. So this is from a species pair. If we find Duquidium pollen, we would call that Logistipollinites florini, and that relates to a genus. So it could come from any one of, I think, about 15 or so genera of Duquidium. We already discussed Terminalia, but as a dispersed, in terms of dispersed fossil nomenclature, we would call this Heterocolpites levigatus. And so that name would accommodate any of those genera, Dactylocladus or Melastoma or Terminalia, all fall within that same species. And if we find Liguliflorae of the Compositae or Asteraceae, all of it would fall into the same species, Echitricolprites spinosus. Our grass pollen, Monoprites annulatus, and our Chenopodiaceae type pollen would all fall in Chenopodipolis multiplex. So these are all species in their own right, species of dispersed fossil pollen, whereas these varying rank from species to multiple families. I hope that's clear, because it's really important for the palynologist to understand that some pollen types are identified very accurately to modern plants, others are not. Others reflect very broad groups of multiple families. Now, for the Cenozoic, my advice would be, until form taxa have been published for each pollen type from Southeast Asia, it's better to use Quaternary nomenclature, because if you try to start using dispersed fossil nomenclature and you don't know, and these species pollen types haven't been properly described, then we'll start losing information in our pollen diagram. So for instance, if we find Calophyllum pollen, which you probably all know, and you call it Roypites species 1 or something like that, then in the diagram, the information from the identification of Calophyllum is lost, and that data set will be of less value to anyone else that looks at it. So just some basic rules on how to name pollen. I hope that's been useful for you. But let's move now to modern pollen rain and pollen transport. I think this is a very important part of becoming a palynologist, is to understand the process of the issues of modern pollen rain and transport. How do we, when we find a pollen assemblage, the pollen assemblage essentially reflects some aspect of the nature of past vegetation. And what we're trying to do is to find out what that vegetation was like and what it might mean in terms of paleoecology and stratigraphy. So how do we establish the relationship between pollen assemblage and the vegetation that produced it? We do this by studying modern pollen rain. And we do this in an area where we know what the vegetation is. And so if we go to an area, maybe we look at a vegetation transect. It might be in relation to altitude from the base to the top of a mountain, or an environmental transect across coastal swamps and say mangrove swamps. We might enumerate the contribution of each plant species to vegetation along the transect. And we might take a pollen sample from the soil, the mud, or moss polsters are very good for trapping pollen from each of those sites. And then we analyze the pollen in the soil, pollen in the sediment sample, and we compare the abundance of pollen with the abundance of the plants that produced it. And in this way, excuse me, we can determine the differential pollen productivity of each taxon. And this is very important because some taxa produce lots of pollen, others don't produce very much. So if we find taxa that produce very little pollen, we have to give it a little more emphasis when we're making our interpretations. We can also make judgments as to the extent to which each pollen type is transported away from its source. And this is also very important in understanding how to interpret pollen assemblages in sediment samples. Now, my suggestion is that every palynologist should undertake their own modern pollen rain study in order to understand how to interpret the pollen record. Although academically people will do this as part of their project. For instance, when I was working in Singapore in the 1980s with Robertson, I used to take people up regularly to the Sedili River, which you saw on the first, on the cover of this presentation. And we would look at the distribution of mangroves and look at and collect samples, and people analyze these. And so they all understood, after all the palynologists working in Robertson understood how to interpret the pollen record in that sort of setting, because they'd looked at the modern pollen distribution in the Sedili River. An area that I studied. When I was a PhD student, I went to Indonesia with John Flenley, and we looked at 62 possible localities across Sumatra and West Java. We were looking for a site that would extend back to the last glacial maximum, so that we could see how the vegetation had changed in relation to time since the last glacial. And we were very interested in finding a locality that was at quite low altitude, about 800,000 meters altitude. So it gave us some idea as to how lowland rainforest was affected by climates in the last glacial. And after looking at about 60 localities, we finally decided that Danau Padang, here in the Kerinci area, that's Danau Padang there, was the locality that we should spend all of our effort on. This yielded about 20 meters of detrital mud, and we thought it was certain to go back to the last glacial maximum. So I set about studying the ecology and pollen rain from this area, and I did this by going to selecting a dozen localities in the rainforests in the area. There were two in the south here, south of Danau Kerinci, in sub-maintained forest. Each consisted of a 0.04 hectare plot, which was 200 meters long, 20 meters wide, and I enumerated all the trees on that particular plot. I then collected 10 samples of soil samples from that plot, homogenized them, and analyzed those samples for the pollen. So I can compare the present vegetation with the pollen that it produced. And I collected those two there. Then we went up to Kayu Aru, collected from about 1350 meters and 1450 meters on the flanks of Kerinci at 1550, up to further on the flanks at about 1700. Then climbed up Gunung Tujuh. This is Gunung Tujuh with a big lake a big crater lake at I can't remember the altitude in meters, but it was 7,000 feet, and collected from 3 more localities at 2,000, 100, and 200 meters. So those 3 localities there are shown on this photograph here, taken from Gunung Tujuh. So my sample 10 was about here. My sample number 11 was somewhere down here. And my sample number 9, probably just below the bottom of the picture here. And site number 7 was over here on the flanks of Kerinci. So I took all the plants that there were several hundred different species of tree. I grouped them according to their pollen types, and made a little diagram of them here. So you can see taxa here that are restricted to the higher altitude forests. These occur throughout the region, and these are restricted to the lower altitude submontane forests. And this is the pollen productivity of the same groups. So you can see that the higher montane trees were higher producers because this is bigger than this. The lower montane trees were low producers because this is a bit less than this over here. In making this diagram, I used the approach of Wright and Patton in a famous paper in 1965 that the pollen sum should reflect the vegetation under study. Now, if I'd have included other pollen into these groups, it would have distorted the picture, so we couldn't make that direct comparison. So this diagram here shows the total pollen recovery in those samples. This is the pollen I used in the sum. This is non-forest regrowth pollen, so we want to exclude that. And this is, these are herbs, these are spores. So I've excluded all of those and just used this group here. And in that way, we can make a direct comparison of the modern pollen with the modern vegetation. And this has been really useful, used it for many, many years. This work was followed up a few years later by John Flennelly and the next research student, Jane Newsom. They climbed Corinchi up to about 3,300 meters, something like that. I don't think they did the detailed vegetation study, but it produced a modern pollen spectrum from submontane forest at about 1,200 meters to the very summit of Corinchi, about 3,300 meters. And this has been the backbone of interpretation of vegetation in terms of climate in Sumatra and Java since that time. Now, just to show how easy it is for anyone to do this type of work, and any modern pollen studies will be useful for you. This is a study recently published, 2020, by Zongjing Cheng and some co-authors. They simply went to some very nice, maybe they did this while they were on holiday, I don't know, but they went to Gunung Jurai, which is a mountain in Malaysia on the coast, very nice place, and collected lots of samples from different altitudes. They did the same on the Penang National Park here, took lots of samples again at different altitudes, and then went to the Matang Mangrove Forest Reserve, which is just down here, which is a place where you've got mangroves and lowland forest occurring together. They put all of those in terms of altitude on the same pollen diagram. And again, you can see this pattern of change from high altitudes through to intermediate, through to the lowlands. It's a really nice study. Anyone can, this is published in a well-known journal, and so it's accessible to everybody. But to have more studies like this from even from Gunung Pangrango, just on Puncak, would be useful. So any modern pollen studies from Indonesia will help us in our ability to interpret stratigraphic and records in the future. So my advice would be for people to think about these when you're visiting. It'd be nice to have a series of studies from across Indonesia in different localities in different settings. Right. Now let's look at the pattern of pollen transport away from a coastline into a marine environment. This is a diagram. It was drawn by Jaizan Jais, who works with Petronas, but it's from a diagram I made about a long time ago. And it shows how pollen is transported down rivers. You have to bear in mind that about 95% of pollen is transported by water. And if we take our sample from the hinterland area here, excuse me, we'd find that most of the pollen came from that local vegetation. But as the river flows through into, say, freshwater swamp here in the upper delta plain, we'd find that if we took a sample from the floodplain here, most of this pollen would be from the floodplain itself. but a proportion would be transported down the river and come from the hinterland. If we move into the lower delta plain where we've got mangroves, again, if we take a sample from the mangrove mud here, most of the pollen would come from the mangroves. A bigger proportion would come from the freshwater swamp, and a smaller proportion from the hinterland. Now, as we move away from the coastline and we take samples from here, of course, here there's no local vegetation producing the pollen. And so the coastal swamp is of limited area. You know, it's got a fixed geometry, fixed area, whereas the hinterland vegetation is very widespread. It cover the whole of the landscape from which this river was flowing. And so as we move away from the coastline, the proportion of pollen from the hinterland will increase, and the proportion of pollen from the mangroves in the coastal swamp will decrease. And this will become more and more prominent as we get further and further away from the coastline. And this is very important when we're interpreting our fossil pollen data. When we get to really deep marine environments, somewhere like this, maybe by this time, if we haven't got turbidites, then the amount of pollen coming down the rivers will be very small, but that 5% of pollen that was transported by air will be much bigger. And so the amount of pollen coming from the hinterland will increase. This process is called the Neves effect, and it was first identified in the Carboniferous by Roger Neves, who was professor of Palynology in Sheffield for a very long time. Right, just a bit about sampling and a few logistics about methodologies. In collecting field samples, always collect your samples, collect far more samples than you actually need, because you'll probably never be able to go back to that place. Each lithophase should be sampled in a manner that would allow you to capture the sedimentology of the unit. So there's no point in taking just one sample from a sedimentological package. You need a series of samples through it. Excuse me. For sequence biostratigraphic sampling, actually closely spaced cuttings are the best. So if you use core and sidewall core samples, they're helpful, but only to augment and to help understand the record that you're getting from cuttings. So close spaced samples in well sections, closely collected samples in outcrop, each lithophase being characterized with sufficient samples to be able to characterize the sedimentology of the section. Now in analysis, of course, log all the pollen and spores plus algae, aquatox, anything else. Take photographs, make sure that these can be circulated between people to make sure identifications are correct. Get good counts. 300 is a good guide, 200 will do very well. But if you've got samples, which you'll understand when I talk about the pollen sum, that are very rich in, say, mangrove pollen, we would exclude those from the pollen sum. And we want to make sure that in those samples that we've got a sufficient pollen within the sum. So if you've got a sample that has, say, 90% mangrove pollen, you want to carry on logging your sample till you've got a minimum of about 100 specimens that are within the non-mangrove pollen sum. And in that way, you'll be able to pick up the hinterland signatures, which will tell you about climate change, as well as the mangrove signatures, which will tell you whether your mangroves are prograding or retrograding. So to present your data, my rule of thumb is to restrict the pollen sum to the hinterland sources, and this excludes the mangroves, spores, algae. They're all logged, but they're excluded from the count. And then they are plotted outside the pollen sum, in which case their representation is really useful for interpreting depositional environments. So the idea of this is not to muddle up the climate signatures from the environment signatures, and that's one way of doing it. Presentation. I always feel that the style of presentation, especially within the Cenozoic, really determines what you can interpret from a data set. And I like to get as much information on a single figure as I possibly can. This is just an example of a well section from the Mahakam Delta. It's all late Miocene to Pliocene in age, and it contains a lot of pollen. So these are mangroves, these are hinterland pollen, these are some markers that are useful for dating the section, and these are spores. Now, the pollen sum, I use total hinterland pollen, which is all the pollen from the non-mangrove coastal vegetation, from the rainforests, from river systems, from mountains, all goes in here. And the fluctuations that we see here are going to reflect climate change or changes in geomorphology of the delta plain, which may be useful in interpreting this succession. Now, to get an idea of the quality of our data, we can plot, this is percentage, plot the abundance here. So this is 0 to 200. So you can see there's huge numbers of hinterland pollen recorded here. So the patterns that we see here are very accurate. The moderately good records up here and moderately good records down here. So the record that we see on this chart from here to here is very accurate. Any event that we see here is probably reflects a transgressive or regressive pulse on this delta. But when we get below here, the pollen recovery in the sum is very low. Notice that these are very low numbers, but our spores are very high. And the reason is that in this lower part, some sort of winnowing has taken place, which has perhaps removed the small hinterland pollen and concentrated the robust spores. So our signals are not very good down here. So we don't want to give this too much attention, but in this section up here, we give careful attention to everything that is going on here. These oscillations here, you can see them in reflected in changes in peat swamp and riverside pollen. These basically parallel the succession of turbidites that we see in fields like Rangas and Gendalo and Ceno in the Kutai Basin. So whatever processes drove those depositional cycles that caused those turbidites also was driving the vegetation on the delta. So if we can devise a model that would explain turbidite deposition and also the successive changes of delta plain vegetation seen in this diagram, then we can get a very good idea of the understanding of what's going on in this particular section. That's a sort of introduction to sequence biostratigraphy that we'll talk about later. We compare that with the broad zonation that you would make just from the age diagnostic species. We've got Dacricarpus imbricatus up here, which picks the Dacricarpus zone. We've got Stenoclena milnei coming through to about here. We can divide that into an interval with low numbers and high numbers of specimens, and I divide those into two different groups, and the interval below is the Florschutzium ruinulalis zone. So this resolution is quite low, but the detail that we see in the mangrove record and in the pollen record of the hinterland is very high, and this varies on the resolution of the reservoirs that we see in the Kutai Basin. So without a diagram like this, it's very difficult to summarize the extremely complicated assemblages that are present in palynology. One point I want to mention is that the different pollen types, you may have 200 pollen types making up this diagram, but I place them into ecological groups, and each group is a group where I think most of that pollen will come from. It needn't all come from that group, but most of it should come from that group. So in this group, I would put things like Barringtonia and Terminalia, but of course, the Terminalia could come from peat swamps. In the rainforest group, I'd put all of those rainforest elements that I'm not, it's a bit of a mixed bag, everything that is not placed in other groups. The Carapa swamps are basically Casuarina and Dacrydium. The peat swamp groups are in here. The riparian taxa riverside things like Pandanus and Myrtaceae go in here. But of course, all of those genera could come in other places too. So we have to realize that these aren't absolutely strict groupings, that things could come from other groups from time to time. And if we do that, then we can generally come up with really good interpretations of these sections. Right, hang on. Let's look at Quaternary studies. Now, whereas the first person to make judgments about how vegetation had changed in Indonesia was Alfred Russel Wallace. He climbed Gunung Pangrango here, which I have also done several times. I recommend it to every palynologist to climb this mountain. It's easy to reach from Jakarta. He climbed it in, I think it was 1854, and he made the first judgment. He noticed when he got to the top of the mountain, you've got montane forest growing to about the edge, the rim of the crater here, but in the crater itself, there's a little meadow. And anyone coming from Europe would immediately be able to recognize most of the plants that are growing in the meadow. There's Edelweiss, there's buttercups, there's primulas, gentians. It's just like getting back to a meadow in somewhere in England or in the Alps. And Wallace noted this, and he was particularly struck on this primrose, and he pondered, how could these plants possibly have reached here, bearing in mind that we're in the south of the equator in the tropics. And he suggested that during glacial times, climates were cooler, and these plants could perhaps have migrated along mountain ridges and eventually found their way to somewhere like this. Partly correct, but not examined further for another 100 years until the probably 120 years until the 1970s, when we started working in Sumatra. Now, unlike geologists working on the Cenozoic, as we heard from Han last week, there were actually some ladies that were involved in palynological studies in the 1930s. And this is Betty Polak, a formidable looking lady. I originally made her picture a bit bigger, but it was a bit frightening, so I reduced the size. She's the person who was responsible for coming to the conclusion that, say, the peat swamps of Indonesia are in fact domed ombrotrophic swamps. She also recognized Rawa Lakbok on the south coast of Java as a very important palynological site. But I don't think anyone has finished this off, and so 90 years later, it's still awaiting study. If someone would be interested in studying that site, getting a borehole that goes through right to the base of the succession, it would make a very interesting study. This is John Flenley. He was my supervisor. I was his first research student. And he undertook a long-term study program in the 1970s. The idea was to study the extent to which the vegetation had changed between the Holocene and the last glacial, to see if there were any similarities or differences between studies that had been undertaken in other areas. And this would have been of global consequence because it was the first study that really, or project that concentrated on understanding the history of lowland tropical rainforests, which of course have our most diverse vegetation anywhere on the planet. So Quaternary studies fall into 4 groups. We can take cores from lake deposits, which was what John Flannery was interested in when I did my PhD thesis. And these show vegetation change perhaps through the last glacial, with an idea of showing vegetation change since then, and also the effect of man on vegetation, say during the last 8,000 years or so. And this was really the first area of emphasis as far as palynology was concerned in Indonesia. Secondly, we can look at coastal and inland peat swamps. Analyses through the peats will show how the peats have formed over time and the nature of the vegetation succession. And information on this is needed to help understand how the peats have developed. And this is important for better conservation management of these peats, because as I'm sure you're aware, the peatland environments across Indonesia are a complete nightmare, and effort needs to be undertaken to conserve these. But also, if you want to conserve them, you've got to understand how they actually developed, and that, I think, is still very not very clearly understood among our peatland specialists. Peat studies also act as analogues to understand the sedimentology of Cenozoic coals, also another very interesting area of study. Then we can look at mangrove swamps, and these will show the temporal development of mangroves in relation to changing sea levels. This is again useful for coastal management and conservation, but for our point of view of stratigraphic palynologists, these studies act as analogues for interpreting Cenozoic coastal plain sediments. And finally, various people, mainly the Australian Monash group, have looked at marine cores, and these tell us about supra-regional vegetation climate change. And they're also useful for interpreting our Cenozoic palynological records. I'll just flick through a few examples. These are some studies from lake deposits. This is Danau di Atas in Sumatra, 1,500 meters, and this is Situ Bayongbong, 1,300 meters. This was studied by Ingelisa Stutz and this by Jane Newsom. And you can see, although these are slightly different altitude, both show the same pattern of change from vegetation dominated by podocarps or conifers to vegetation dominated by oaks, such as this. Altogether across Indonesia, there's probably not more than half a dozen localities where profiles like this are available. And so if we want to understand how the climate of Indonesia has changed since the last glacial maximum, which is important for ongoing climate modeling and understanding how the climate's going to change in the future, then more studies like these would be very useful. Now, the sites that were studied in Sumatra and Java are shown here on a diagram that shows altitude here and age along here. And each of these sites is, each of these horizontal lines is a palynological analysis of a lake deposit. So the one I studied was Danau Padang, and it's the lowest. The idea was to study the boundary between the lower, lowland rainforest and montane rainforest. What this diagram tells us is that these data sets here are from New Guinea, that during the last glacial, high altitude vegetation changed much more extensively than intermediate altitude vegetation. You might say, oh, simple. It's just a different lapse rate during the last glacial, but that doesn't actually work. Climatologists are not happy about such a statement at all, so we've got to find an ecological reason as to why the high altitude vegetation changed much more extensively than the intermediate and low altitude vegetation. This is something that people need to work on. That's why we need more data sets that extend back to the last glacial maximum. Now let's look at peat swamps. Again, it's something that I've been involved with many years ago. There are 4 different types of peat swamp in Indonesia. We have what I would call deltaic ombrotrophic peats. This is like the Marudi peat swamp in Sarawak, which was studied by Anderson and Muller. This is these are watershed peats, or watershed or karapa peats, as I would call them, such as Sembangau in South Kalimantan. This is a topographic planar peat, as in the Burbak swamp of South Sumatra. And this is a topotrophic inland valley peat. This is from Malaysia, but we'll find similar examples in Indonesia. And these are much smaller scale. Now, people working, it seems to me, people working on conservation or understanding the present day peatlands don't really put in enough geological thought into their models. And basically, these peat swamps have formed after sea levels stabilized about 5,000 years ago. The Sabango peats formed much earlier than that, when sea levels were perhaps 50 or 100 meters below the present day. And they simply formed because rainfall increased and the amount of rainfall was much higher than the amount of runoff. So these areas became waterlogged and peats developed. We'll look into this one in more detail in a minute. The Burbak swamp in South Sumatra is actually in a subsiding area. So most of the peat is preserved below the present sea level. As a result, this has got a more or less horizontal surface and is planar rather than umbrotrophic. And then Tasitbara is a topotrophic valley peat, of course. So let's look at Sabango because it's a really interesting section. Oh, but first of all, the vegetation on peat swamps. At the edge of the peat swamp, we'll find mixed swamp forest, which is similar to lowland rainforest. As we move into the center of the peat dome, the peat gets thicker, and we tend to get a single species dominant. And this is this vegetation is divided into phasic communities. So we go from phasic community 1 at the edge to phasic community 6 in the center of these peat swamps. So this here we get a dominance of Shorea albida in peat swamps in Brunei. Here's a tree of Shorea albida, very large trees. Then as we get higher up on the peat swamp, the amount of nutrients decreases, and we get pole forests, which are characterized by very small trees like this. And then when we get onto the sort of summit of the peat, we see this open padang, it's called. So this is the Katina, from mixed swamp through to padang. Now there's two other peat types here. This is Karapa peat in Brunei, and this is dominated by Casuina with the umbrella, or Gymnostoma. This is Gymnostoma nobilis. And this tree here is Dacrydium. And so these two are clearly associated. And we can see this in the fossil record. So this is an important association to bear in mind. We'll find this association right back to the Oligocene. And these are topotrophic peats dominated by pandans. And I just want to run through the diagram that I produced 30 odd years ago from the Sabango peat here in South Kalimantan. I didn't run any radiocarbon dates on this because I hadn't got a budget when I did this study, but this was later dated by Susan Page and other people at Nottingham or Leicester University. And what they found was that there was a nonconformity, a break in here about 6,000 years. So our peat started to form on, say, grassy open swamps about 10,000 years ago. It developed through mixed swamp forests and into a phase of Karapa peat with Dacrydium. And then finally, it developed an interval at the top dominated by Combretocarpus and Calophyllum, like this. Now, if we look at that in cross-section, you can see what's happening here. This peat started to build up when the sea level was 50 meters below the present sea level. So this is this peat accumulation has nothing to do with sea level change. Initially, we had mixed swamp forest growing here with Campnosperma and things like that, and then that gave way to the Dacrydium forest here. And then the peat stopped growing 6,000 years ago, and this became a deflation surface. So for the next 5,000 years, no peat formation. Then as sea level stabilized about 5,000 years ago. Then peat started to build up in the Sabangao River and just prograded up the river system and eventually onlapped this peat dome. So this is a young deltaic peat. So this is Karapa or watershed, this is deltaic. And the deltaic peat onlapped over the older peat. So you can see that these peats are very complicated. And if we want to understand how they're formed and how to manage them, we've got to understand a lot more about their history, which would require a lot more palynological analysis. Now, one of you, Eko Yulianto, has done a little work on this. He looked at a core quite close to the locality I worked on, just up here by the river that I can never pronounce, Kahajan River, and he identified, or his core was quite covered the same interval as this section here, and he did a much more detailed analysis than me, but he saw these 3 successive phases of development of peat. Now this is Crataxylum, which is a tree that is characteristic of regrowth and burning. So what this suggests is that we're picking up here 3 phases of renewal of peat development. And we can mark those on like this. And if we look more carefully, we can see that we have a little peak here of Neoscordacinia, and that also includes Daquidium. And what is happening here is, if we put on Eko's charcoal analyses, we can see that at this point, this peat swamp was probably burnt by some reason, left lots of charcoal, and then the Crataxylum developed over the burnt surface of the peat, and then eventually it was replaced by Karapa mixed swamp forest, which is equivalent to what we're seeing over here in my work about this particular point. So we've got some very nice radiocarbon dates over here. And so through Eko's work, you can actually see that this section of peat swamp took about 250 years to change from a burnt out peat swamp to a nice mixed swamp forest. This sort of information is really important for understanding how to manage modern peat swamps. And if this wasn't originally noted in the paper, but I think this is the most important part of this study, that it shows that palynological analyses can actually provide guidelines for undertaking, understanding how to manage and how to develop these peat swamp areas. Right, another of Eko's studies here that I thought I'd pick this out as a really interesting section that goes through a coastal mangrove swamp. This is from Sulawesi. He took lots of cores here and studied this one here, I think it is this one, and he analysed that for palynology. Now, the interesting thing about this particular peat is that this particular profile is that the mangroves are growing on peat. And today, if we look around the mangrove swamps of Indonesia, hardly any of them ever grow on peat. So a very interesting subject coming from this paper that would be worth developing because it's relevant right at this minute is why do mangroves sometimes grow on peat, and why do they usually grow on muds? In the rock record, we can find lots of mangrove pollen occurring in coals, and we need a model to explain why that is the case. So these studies, preliminary, can be developed, and new studies will really help us understand more about how to understand the fossil record, and also help to understand how our coastal mangrove swamps have prograded during the course of the Holocene. Finally, in the Quaternary, I just wanted to show you this record from a deep marine core. This is from the Lombok Ridge which is a deep marine. I think I'm not sure of the water depths, probably about 2,000 meters or something like that. But it's yielded quite a lot of pollen, and it's been analysed for oxygen isotopes, and from these we can interpret the stages of the marine isotope stages through the late Pleistocene. And we can see the last glacial and the penultimate glacial. But if we look at the pollen record, we can characterize each of these packages based on pollen alone. So the glacial stages dominated by grass pollen here and here. We can see the glacial terminations characterized by appearance of mangroves here, 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 and here. We can see the interglacial intervals characterized by abundant spores here and here. Just from this little diagram, we can identify systems tracts through, or their lateral equivalents, in a really deep marine setting. So these studies really helpful for us to understand how to build models to interpret sea level change in the geological record. Well, we'll leave the Quaternary there and have a look at the Cenozoic. What I want to do here is discuss how to build a palynological zonation for the Sunda region using age-restricted palynomorphs. Now, this actually has been a major challenge for a number of reasons. All the early work in petroleum exploration was on very young sections in Brunei, Sarawak, mainly the late Miocene and Pliocene, and very little was happening there. And so it was quite difficult to work out a detailed zonation at the beginning. The other problem is that the Paleogene stratigraphic record is highly fragmentary due to the number of tectonic events that have affected the region. And to build a comprehensive zonation, we need data sets from widely separated localities. However, once we have this succession in place, we can use the events to characterize and explain the very tectonic events that those breaks within the rock record. And we can use the identification of pollen types that have dispersed from other plates to help understand exactly what was happening as we move from say the early Eocene into the middle Eocene, things like that. Excuse me. Now, for more detailed age interpretations, especially in the Neogene, we really need to look at a different approach using sequence biostratigraphy, because our age diagnostic palynomorphs are not going to give us the sort of resolution we need. And I'll explain how we approach the identification of palynological zones in the Neogene later. But first, let's look at the Paleogene. Now, this is a diagram that I first presented in Utrecht in a workshop that I gave that we ran for about 3 years. We called it the Utrecht Pollen Spore Masterclass. And I've tidied it up a little bit. But basically, I divided the succession into zones, and I named these zones, gave these the prefix P or E, depending on whether they were Paleocene or Eocene. Now, we can't see all of these zones in any one location. We can only get this zonation when we bring together data sets from different places. And the data sets used are from the East Java Sea for the early Paleocene, Sarawak for the late Paleocene, probably also some of the early Eocene. We then have a big break here with the Sarawak orogeny and a long process of rifting taking place, which has affected the whole area. And then after that, to get the story through the Middle Eocene, we need to go to Sulawesi to get the earlier part of it. That may just extend into the early Eocene, but not very much. So I tend to call all of this Middle Eocene. The major part of the Middle Eocene is reflected in the Nangulan Formation in central Java, which has been studied by Dr. Lilono, who is director at the Geological Survey in Bandung, and produced this local zonation, which is useful for correlating within the Nangulan Formation itself. And then afterwards, we look at the late Eocene in the Tanjung Formation from East Kalimantan. We can also see this, but not so well dated, in the Baya area in southwest Java. So to get the zonation from the Paleocene through to the Eocene, we need to go to 5 separate localities: East Java, Sarawak, Sulawesi, Nangulan, and Tanjung. So let's just look at the early Paleocene, I think, from the pre-Nimbang Formation. This is from subsurface sections from the East Java Sea here. Now, the pre-Nimbang is a bit of a hodgepodge. It's got sediments of several different ages in there, but probably the youngest one is rich in palm pollen, especially this trichotomer sulcate type, which we were going to publish this about 20 years ago, but the company we did this for eventually was taken over by another company. We lost contact and it never happened. But we have all of this work ready for publication, but unfortunately, because of the issues regarding company takeovers, we've never actually published this. But this is we've called this Trichotomer sulcites javanis, and we intend to publish this name very soon. This also occurs with a proteaceae-like pollen, which we're calling Proteacidites sigalii. This is one from West Africa. You can see they're very, very similar. And this is restricted to the Maastrichtian through to the probable early Paleocene. This we also see in the Paleocene of Myanmar. And so if we bring these together, the likelihood is that we've probably got some early Paleocene or Maastrichtian here in the East Java Sea. And these are the oldest sediments that we see, the oldest Cenozoic sediments that we see anywhere across the Sunda region. At the top of the Nimbang, the pre-Nimbang, we have a big break. And after that, we see pollen types that have affinities with taxa we would expect to find in the Paleocene and early Eocene of India. Things like Palmipollinites cochensis, Lachiopolis ovatus, Linagiopolis, even Marginipolis. All of these have probably come from India as a result of the collision of the India plate with Asia in the Eocene. So, but we'll talk about these again in a minute. First, I want to go to Sarawak and look at the Sarawak succession, which comes from a paper by Jan Muller, published a very long time ago in 1968, but fantastic data. The only problem with Muller's work is it needs to be tidied up and put together so you can see what's going on. Also, in his original age interpretations, he overestimated the age, and so I modified the ages in a paper I wrote about in 1998, and I think most of his what he is now calling the Cayenne Formation is a Paleocene age. A student at Royal Holloway did some work in this area and did some radiometric work on these and also some zircon analyses, and also came to the conclusion that most of these sediments are Paleocene and early Eocene. Now, Muller divided these into the Rugu bivesiculites and the Proxpatites zone. Rugu bivesiculites is a gymnosperm of northern hemisphere origin. It's really common in these sections, together with what Muller called allisporites, which is really pine pollen. And so to have such abundant pine pollen, but abundant conifer pollen, what does this tell us? Well, the student I was mentioning, Tim Brightfield, he showed that these were alluvial fans coming from the Schwaner Mountains. And these reflect the unroofing of the Schwaner Mountains. And it suggests that the Schwaner Mountains were probably 3,000 meters high, something like that, to produce so much pollen that that it can be reflected in this manner. So I've called this zone P3 to fit in with the P1 and P2 from East Java Sea. And then this is replaced by the Proximatites zone in the late Paleocene, and this is dominated by Spinizanocopites echinatus, and also by Aquila pollinates, very unusual, and also Apocynaceae pollen, Psilodiparites wolfendeni. So these are very distinctive assemblage, and this represents the end of the Paleocene. And then the early Eocene, also dated radiometrically, is characterized by Retitriparites variabilis, and also Discoidites. Brown loria pollen comes in at the beginning of the Eocene. And so you can see a very distinctive succession, and very different from what we see in the Nangulan and other Eocene successions from Indonesia. So this is from a well section analyzed by Lemigas. Thank you. And it contains pollen types that most likely came from India. Things like Parme pollinites catchensis and other Parme pollinites species. These occur right at the bottom of the section, basically at the base of the rift succession. And for these to be here at this time means there must have been contact with India. There wasn't a physical contact in terms of land connection, but India and Sunda were in the same latitudinal zone, and so these taxa were able to disperse across into the Sunda region. This appearance of Indian taxa was quite gradual. So first of all, we see Parme pollinites, and then later we start seeing other taxa like Lachiopolis, which is Durio. All of you in Indonesia probably love to eat durian, and if it wasn't for the India-Asia collision, you wouldn't have had any durians to eat, because they would never have found their way into Indonesia. We then find Marginipolis coming in the middle of this zone, which I'm calling E3. And then later on, we find another pollen type we find in here is Restionidites punctulosus, which is from the family Restionaceae. This is very common in the early part, but becomes less common later on. It's replaced with Parme pollinites, you can see here. And this suggests that the climate has gone from more seasonal to much wetter. And this produces this, together with the appearance of wet climate indicators like Retistephanocalpites williamsi from Cenozoic, Gonistylus, and things like this, tells us that we're changing from a more seasonal to a much wetter climate as we go from zone E3 to E4. And I put some approximate dates over here. Now if we move over to the Middle Eocene, this is taken from Dr. Lilono's thesis, from who's now director at the Geological Survey in Bandung, and he did a very detailed study. If we look through this section, we can see a relatively uniform succession of assemblages, but we see some gradual changes in the upper part. We see Podocarpus coming in within what I'm calling zone E6. And we see a change, concentration of wet climate elements in the middle Eocene here. And on the basis of that, we've called this E4, E5, and E6. So this succession, it provides a picture, sort of the intermediate sixth part of the Middle Eocene. Several taxa here need to be described. This is Margocolparites species 1, which is this one here. And this is Baccu tricolporite species 3 from Dr. Lilono's thesis. These are all really useful age diagnostic taxa, and we need to get these published at some stage. Moving up into the late Eocene, this comes from the PhD thesis by Duncan Witts. This zonation has been published in Journal of Asian Earth Sciences. But basically, we can carry on from E6, which has our first appearance of Podocarpus. E7 is based on the first appearance of Cicatricoses spirites duragensis. E8 by the first appearance of Meiripolis neocatensis, and E9 by the first appearance of Magnostriatites howardi, or Ceratopteris spores. This zonation, we're becoming very confident with this. We think we can interpret this in several different localities. This succession doesn't go to the top of the Eocene. The Eocene is much higher. So we think that the Magnostriatites howardi appearance is about 35 million years. The appearance of Meiripolis, we think, is about 37. But recently, a student working in Amsterdam, Hashweng Huang, has been looking at this section in Myanmar, and we think this zone boundary should be pushed down a little bit. So that will all be coming out and published quite soon. And so those are the criteria that I've used to build up this zonation. We can see the pre-Nimbang, we can see the succession from Sarawak. We can see the effect of the orogeny and the onset of rifting coinciding with the collision of India with Asia, and that allowed all of these elements here, which are all come from India, and the result is a zonation that extends from the throughout the Paleocene to the top of the Eocene. Now, if any of you are looking at sections that might cover this interval, you might find something that sort of doesn't fit in here. Maybe it's an extra section that we've never seen before, but please bring it to my attention because it may result in slight modification of this zonation. Now, I want to put this slide in, which shows the effect, characterizes the Eocene collision of the Indian plate with Asia and the effect that that had on pollen assemblages. If we look at the palynological record of the species that I've been talking about, they're shown here, and the ranges are in grey for the Indian plate. These have all been determined by palynologists at the Birbal Sahni Institute of Paleobotany in Lucknow, and there's a very active group there currently working on this particular section, especially on the early Eocene and the Paleocene, which are very interesting for all of us. I put on the different changes in climate here with the increase in the interval with common the common seasonal pollen going into more common humid pollen. Now, interestingly, from some work that I'll explain in a little while, we think that the land connection was formed at about 41 million years. So after 41, plants could migrate from India to Asia without having to cross a water body. All of the plants that are represented here up until 41 must have had to have crossed a water body to get to Southeast Asia. But after this time, things like Meiripolis and Ceratopteris seem to have the same range in India and Asia, so they could easily make that movement. Now, I've summarized the different positions of the plates here. This is the Paleocene, where India was probably too far south to allow dispersal. In the beginning of the Middle Eocene, about 48 million years, we see the first dispersals. About 45 million years, we see lots of dispersals. When I wrote this paper about 2 years ago, we thought there were some dispersals in the other direction, but now we've found that that's not the case. So forget that little arrow there. By the time we get to the late Eocene, we can see dispersals in both directions. And then based on molecular work, after that time, we think most of the dispersals were just from Southeast Asia into India. Now, to date this succession, and also to understand these dispersals, we've approached this from a completely different perspective by looking at the molecular record of plants and animals that occur in both India and Asia today. And I did this together with a colleague, Sebastian Klaus, basically we took all of those plant or animal species that have been sequenced using molecular methods that occur in both India and Asia, used whatever available molecular clocks to determine the age of these dispersals, and then made two little graphs showing dispersals from India to Asia, which is the blue line here, and dispersals from Asia to India, which is the yellow line. Now, if things were absolutely uniform, both these lines would be straight, but that doesn't actually happen. What we see is that at 48 million years, there's lots more dispersals from India to Asia, and very few dispersals from Asia to India. And then at about 40 million years, or 38, we find that the dispersals from India basically stop, but then we see lots of dispersals from from Asia into India. And I explain this by suggesting that when India was, during the Eocene, it was in the same latitudinal zone as Southeast Asia, and its aggressive flora was able to colonize the Indonesian region here. But by the late Eocene and getting up into the Oligocene, India had drifted northward into the northern hemisphere high pressure zone, so it developed a strongly seasonal climate here. Asia, on the other hand, had always been in this position, so it had already a rich and diverse seasonal flora and fauna, and that was able to then disperse into India and make appropriate colonizations. Hence the change of direction from India to Asia when the climate, with wet climate elements, from Asia to India for dry climate elements. This also has the benefit that it helps us, it's based on a completely different method of dating. And so it really helps us to date the timing of those Indian dispersals at about 48 million years, which is agreed here based on the molecular work. For the Neogene, this is Jan Muller, who devised the Florschuttia zonation, which is so useful in the Miocene. I tried to tidy this up about 30 years ago in a paper in the Geological Society of Malaysia. My approach in the 90s was, well, if we identify more and more pollen types more accurately, we should be able to get more and more zones out of this. But then I started realizing that that wasn't the case, and that most of what we were seeing in assemblage changes related to what I would call sequence biostratigraphy. And so I've tried to now concentrate on looking for those events that we're absolutely certain have specific age diagnostic significance. And there's only a few, but if you've got really good datums that you can pick across a wide area, those are very, very useful, rather than having lots of datums that are all rather inaccurate. So the datums that I find most useful and that we can date quite accurately now are the first appearance of Florschuttia levipoli, which falls within planktonic foraminiferal zone N4. It's with Globigerina kugleri, and it falls within depositional sequence VIM32b. These are the sequences, the sequence biostratigraphic packages that we'll be talking about in a little while, in about 10 minutes time, 5 minutes time probably. So the first appearance of levipoli in VIM32b. Then we find that Florschuttia trilobata has a big reduction in abundance. Actually, this is also within the top of the range of Globigerina kugleri, and it falls within cycle VIM34. So with these two events here, we can independently pick up VIM34 and VIM32. And if we look for cycles that are driving these, then we can fingerprint those cycles with these two events. If we move up further into the later part of the early Miocene, we find the first appearance of Florschützia meridionalis. This is very nicely dated by reference to nanofossil zone NN3. This coincides with planktonic zone N6, probably N6 and N7, but this falls within VIM depositional cycle VIM42. So picking up the base of this picks up VIM42, and that actually is a very useful regional pick. Then as we move further up, we find, if we look at the record of Florschützia semilobata, now this is, you could call this a sort of top continuous, because about this point, Florschützia semilobata disappears, but it does sort of come back again in the later part of the middle Miocene, about here. But we'll not worry about that for the moment. We see this sudden disappearance of semilobata, and it disappears exactly the same time as the nanofossil Helicosphaera ampliaperta, and that identifies depositional cycle VIM53. So a very useful pick, and remarkably, we can find this in very, very shallow order sections with the ampliaperta coinciding with semilobata. And the same with Florschützia trilobata. Although we will file records of trilobata in younger sections, there's always a big reduction in abundance. It coincides with, it falls within nanofossil zone NN5. If you're lucky and in a really deep order section, you might find some preobulinas in it too, and that will tie it with VIM cycle 54. So these picks, if we can integrate those with foraminiferal data, will be really helpful for helping to calibrate our depositional cycles through the early and middle Miocene. Then we get up into the late Miocene, and we see the first appearance of Stenoclena milnei. That comes in VIM about 64. A little bit of error on there, but it's fairly close. And then we see the increased abundance that we saw on that chart earlier on, and that comes in VIM 74 here. The extinction of Stenoclena milnei type is at 1.5 million years, and that falls up here in the Pleistocene. The other datum that we can use is the appearance of Dacrycarpus imbricatus. I've spent many years trying to work out exactly when this happened, and the place where it was originally picked was in the Barham Delta. I've recently been fortunate enough to work on the Barham Delta, and we find that it falls within VIM 86. That's within nanofossil zone NN15. So just within the upper part of the early Pliocene. So that's about the framework for the late for the Neogene, looking for specific events that we can correlate across a wide area, rather than lots of more detailed events that may be all rather poorly picked. So to look further, we need to start thinking about sequence biostratigraphy. Now, as I mentioned earlier, sequence biostratigraphy is the process of identifying depositional sequences from the examination of microfossil distribution patterns. Now, there are a lot of benefits over traditional biostratigraphy. One, it provides much higher resolution. Secondly, it utilizes, and I think this is really important, it uses all of the techniques of the Quaternary palynologist. And these are often overlooked by stratigraphic palynologists. There are lots of methods that used in the Quaternary that stratigraphic palynologist tends to forget about. And actually, it utilizes all of the biostratigraphic information, whether it's foraminifera, nanofossils, or palymorphs. All of it contribute towards building up our sequence biostratigraphic model. Once we've made a decision as to what constitutes the particular depositional cycle, we can then try and correlate those, and they relate to geological surfaces. We can tie it directly to seismic. If we have good seismic, we can convert our biostrat to two-way time and cross-check our data sets directly against the seismic, loop around and make sure that we haven't got any misties. If we have, go back, reevaluate our data and find out where the error was, because all of these, everything should theoretically fit together. There's either a mistake in the seismic or there's a mistake in the biostratigraphic interpretation. What I've found typically is when undertaking a regional detailed evaluation, our first pass biostratigraphic interpretation has got some mistakes in it, but the seismic interpretation will probably have some mistakes too. And we usually find it's about 50-50. Half of the, if we adjust some seismic picks by a reflector or two, then we'll find gradually that everything falls into place. And in the end, the geophysicist will be much happier with his succession of picks. Now, sequence biostratigraphic packages almost invariably relate to glaciostatic sea level events. And if we can understand these, it makes it a lot easier to understand the influence of tectonics on sedimentation, because we can separate out the events that clearly relate to glaciostacy and those that relate to tectonics. And finally, it allows you to determine sedimentation rates, and you can map these out, you can compare them. It provides a new perspective on how to interpret biostratigraphic data. Now, I want to illustrate these with 4 studies. One is, first I just look at the model a little bit. Then we'll look at the Namkhon Son Basin, which was the first place where we made a really serious publication using this method. Then we want to look at the Kulong Basin, recently published last year, where we got a very high resolution stratigraphy for the Oligocene and late Eocene, which allows us to accurately date all of the several unconformities within the Oligocene. And then finally, I want to look at a Java man locality where we use the same methodology to produce a really precise date for the appearance of Homo erectus in the Mojokerto area, which was, we heard about last week by Han van Gorsel, was discovered by Van Koningswald in 1934 or something like that. So let's just look a quick look at what sequence biostratigraphy is all about. And for a little while, we'll talk about foraminifera. So if we have a shelf setting here, we get a transgression and then a regression that will result in a distal locality. You'll see the number of foraminifera per unit weight of sample increase and then reduce. In a very distal setting, you'll get lots of planktonics and some benthonics. If you're in a more medial shelf setting, you might get a peak of planktonics at the maximum flood, but otherwise maybe more benthonics. And if you're in a very proximal setting, we might just get a maximum of benthonics and no planktonics at all. Now, if we have a marker taxon in here, which for the benefit of the discussion, became extinct at the end of this cycle, it could occur to the top of the succession here, but maybe it would only occur in the maximum flood here. And so we could say, oh, well, this correlates with this, but that isn't strictly the case. Actually, these are diachronous. And it's only by modeling this with a sequence biostratigraphic approach that you can make this really clear. So the point of correlation is the foraminiferal abundance maximum below the top correlates with the abundance maximum coinciding with the occurrence here. Now over here, based on the traditional approach, we probably wouldn't date it. But if we could convert our data sets to two-way time, compare to seismic, we would say, this little acme of foraminifera here is the same event. We can correlate that across. Now, maybe there's some reworking in this section too. If there is, it's probably at the transgressive surface down here. Now, if we approach this from a sequence or without thinking about sequence biostratigraphy, then we might think, oh, these could be in situ. So there must be an unconformity in here somewhere. And look, they tie with this occurrence over here. Maybe this section here is a lot older. But from a sequence biostratigraphic perspective, this is in a poorly fossiliferous interval. So these are suspect. If these were really in situ, they would be expected to occur in the maximum flood. So this way, by looking at the pattern of abundance of microfossils, we can make a judgment as to what's in situ and what's likely to be reworked. So where does the pollen come in? Well, this is the medial locality as far as forams were concerned. What we would see is a big mangrove pollen abundance maximum would come in directly below the maximum flood. This would reflect the transgressive systems tract and would be dominated by Rhizophora pollen up here, as opposed to the back mangrove taxa, such as the Florschutzias. So looking for a succession where we see an abundance maximum of mangroves with Rhizophora, followed by a maximum of foraminifera, picking the maximum flood. Picking that couplet is a really important judgment based using towards building a sequence biostratigraphic story. We might also see a change in the climate suggested from hinterland pollen, from more seasonal during the low stand to more per humid at the time of the maximum flood, and then reducing again during the late high stand. And if we were to calculate the number of specimens per unit weight of sample, we'd find a minimum at the maximum flood and high numbers down here in the low stand or in the transgressive systems tract. These are just a few of the examples. There are many more, both with foraminifera and palynomorphs. But you can begin to see that if you look for these patterns, you'll eventually see a succession of patterns that allows you to identify a sequence. So this is a summary diagram. Actually, this is a bit outdated now because we have a paper coming out, we think, shortly, which will replace this, but it basically gives the same story. If we have a complete low stand and high stand package, then the low stand, we would expect to see basin floor fan, slope fan. We might see secondary condensed sections between those fans, and they might mislead us as to what is a real primary condensed section and what isn't. But if we look at the climate record, these should have a different climate signature to those from the high stand. So that should allow us to separate the low stand secondary condensed sections from the primary condensed section up here. We also have the mangrove pollen abundance maximum. If we have low stand deltas, we would expect those also to produce mangrove pollen maxima, but most likely those will be dominated by sonratoid mangroves, not the Rhizophora mangroves that occur in the maximum flood. So we can tie these into the Veil model, such as this, and before long, we can duplicate this model without any reference to lithologies or to seismic, and then we can use that as a cross-check for the interpretations based on lithology and seismic. So let's look at some examples. The first serious publication was on the Nam Con Son Basin in Vietnam, published in the IPA. We'd previously done work on East Kalimantan in more marine sections, where we'd named our cycles after the abbreviations of nanofossils like Discoasta, Tamalis, and Sphenolithus abies, for instance. But we couldn't use these in marginal marine sections, as you might find down here. And so difficult to apply these terminology throughout the record. Then we did some work in the Malay Basin, where we interpreted our sections in terms of Malay Basin seismic group subdivisions. But people working outside Malay Basin didn't like this, so we came up with this notation, VIM. VIM means Vietnam, Indonesia, and Malaysia. So these zones, or these cycles, apply throughout the area. By integrating the foraminifera and the palynomorphs to identify the cycles, and then fingerprinting them with planktonics and with nanofossils, we produced this time framework here, and these VIM divisional cycles indicated here. These have slightly been modified, but basically very little has changed in the last 10 years. Just to give you some idea as to how these are characterized, the Pliocene cycles, Pliocene, late Miocene, solely on nanofossils and foraminifera, so we'll ignore these. When we get down into the middle Miocene, you can see the cycles very nicely reflected here in the gamma log. Then we find the Florschuttia meridionalis coming in here together with Catapteryx dissimilis, indicating VIM42. We see Florschuttia semilobata coming together with Helicosphaera amplioperta, indicating VIM53. We've got the top of Praeobulina coming just below the top of Florschuttia trilobata, and this is VIM54. So with this succession of events, we can fingerprint these cycles and eventually come up with a framework that is repetitive and can be cross-checked against seismic. For the early Miocene, we're much more reliant on palynology alone, and our successions with just a few shallow water foraminifera coming in. We can see, for instance, in VIM32b here, we've got a maximum of mangrove pollen dominated by Florschuttia trilobata, followed by a little maximum of rotulid foraminifera. And then here we've got freshwater algae, followed by a mangrove pollen maximum, again with abundant Florschuttia trilobata, followed by a peak of larger foraminifera. So you can see that cycle, cross-check it against the gamma log, and we've got a depositional cycle. Then here we get the same sort of pattern, maximum of algae, followed by a maximum of mangrove pollen, and then we get a maximum of agglutinated foraminifera, again picking a cycle, and that identifies, in this instance, VIM36. And then at the top, again, a maximum of Magnostreaotites howardi, indicating a lacustrine facies that are filling in, followed by a mangrove pollen maximum, and also with a rotulid foram abundance. So you can see that the cycles are very distinctive. We can use, in this instance, the abundance of Florschuttia trilobata to fingerprint this cycle. This occurs below VIM42 over here, and so we can interpret the remaining cycles by the stacking pattern. So altogether, we can use this approach to produce a really high resolution stratigraphy for the Nam-Conson basin. The succession as we see it at the moment is presented here, and we have a paper coming out soon that gives a lot more detail about how each of these cycles is characterized and identified, hopefully in another few months. Let's look now at a completely different scenario in the Oligocene and late Eocene of the Coulomb Basin. Coulomb Basin is this just on the northern, sorry, on the just offshore the Mekong Delta. It's a major oil producing province. It's been drilled with thousands of wells. Detailed palynology has been done on lots and lots of sections at very high resolution, but has never been put together until our evaluation a few years ago. This is all published in a paper in Paleogeography, Paleoclimatology, and Paleogeology. And the section we're interested in is the Oligocene, which goes from here to about here, and the late Eocene, which is the basal part just down here. This includes the Syn Rift, of course, and Post Rift, and then we see some marine influence coming in at the very base of the early Miocene, and but other than that, this section is entirely non-marine. Now, if we analyze a well from here for palynology, we'll probably find that it's really full of freshwater algae. I mean, huge numbers. So if we plot the freshwater algae in terms of total palynomorphs, they'll give a spectrum something like this. So you'll see lots of freshwater algae in the lower part, not so many in the upper part, like this. But this is quite difficult actually to see what's going on, because we're mixing up the pollen and the algae. And that's a mistake. Always keep them separate. If we plot the algae outside the pollen sum, outside the hinterland pollen sum that we talked about earlier, then the algae show this sort of pattern. Lots of really sharp and distinct depositional cycles. Now, I've plotted on here also the number of specimens per slide, and you can see that these are very similar to the number of specimens per slide, but this assumes that numbers of specimens per slide has been recorded. If we plot simply outside the sum, then our cycles are also represented outside the area where our data is poor. So the number of polymorphs will reduce as you get in the base of the well due to thermal maturity, but the signals are still represented. So by plotting the algae outside the sum, we can see the cyclicity of the algae. I've plotted on here the fern spores as well, and in some cycles these are important, and these are lake margin pollen and spores. Sometimes they're helpful, but in the more distal facies, it's always the algae that are most important. Now, these cycles, there are lots of tectonic events in this succession, and so we're not going to see all of the cycles in one particular well. So here we've got several wells. We can correlate each particular well from a different locality, and you can see that there are a lot of cycles, altogether about 36 from S2, which is just below here, up to S31 up here. There are some A's and B's, and altogether we have about 36 cycles. Now, what is actually driving these cycles? That's something we need to ask. Well, some cycles have been analysed in great detail. These are some cycles that were analysed at 5-metre intervals using cuttings analyses, and they give us a very interesting story. We can see the algae maximum, 1, 2, 3. So these are sort of lacustrine maximum flood events. Then we see some spore maxima, and these are Magnostriatites howardi, and I think these spores are coming from floating vegetation mats on the lakes, and so they always come a little bit after the algal maxima. But again, we've got 3, 1, 2, and 3. And then if we look at the lake margin vegetation, which in this instance was mainly acrosticum, and you've got to bear in mind that although you think of acrosticum as a mangrove fern, it hasn't always been like that. There are lots of studies that demonstrate that acrosticum grows in completely freshwater floodplain settings, and this was the case in Vietnam, definitely during the Oligocene. So we can still see our 3 cycles, 1, 2, and 3. So we go from open lake to floating mats to prograding lake margin vegetation. So what was the hinterland vegetation telling us? Hinterland vegetation is plotted here, and we can see changes from more seasonal to more wet climate. Seasonal, wet, seasonal, wet. So these cycles are all driven entirely by climate change, and those cycles will be driving glacioceustacy in marine sections. We can see the change in climate over here. How do these events all tie to seismic? Well, we convert our data here to two-way time, and then we follow this across, and if we look at our seismic line, we find that the algal maxima, 1, 2, and 3, correspond exactly with the high amplitude seismic reflectors. So what does this tell us? It tells us that the biostratigraphy is exactly matching the seismic. It also tells us that our seismic interpreters, our geophysicists, are seeing events here that are almost all driven by glacioceustatic climate-driven events. And so geophysics is actually giving us a paleoclimate signature too. Probably geophysicists might not like to hear that. So these cycles are all driven by glacioceustacy. Now, to make sure that our correlations are accurate, we always cross-check, take each adjacent well, convert to two-way time, and cross-check against seismic, loop tie until we've ironed out all of the errors. And by the time we've done this, we've got a really tight correlation around the region. What's actually driving these cycles? Well, they're so regular, it looks as if it's an astronomical driver of some sort. So I looked into the different astronomical cycles that might be driving this, and there are three that are possible. 1.2 million year tilt-driven cycles, which are reflected on this diagram with this curve here. Now, if these were driving the cyclicity, then the sediments at the bottom of the well down here would be Paleocene, and that's simply not the case. So we're not seeing this. There could be 100,000 year cycles, as we see in the Quaternary. Well, if that was the case, the sediments at the bottom of the basin would be late Oligocene, and that's definitely not the case. So what is the alternative? The alternative are 405,000 year eccentricity-driven cycles. Well, you'll remember when we looked at the Tanjung Formation, we found the oldest occurrence of Ceratopteris spores, that's Magnostriatites howardi, at about 35 million years. And they occur in flooding events in cycle 6. And we've got the Oligomycene boundary picked here on the extinction of Cicatricosis brightes daragensis and a couple of other characters. Between here and here, I think we have 31 cycles, and we only have to do some maths, and that tells us that those cycles are about 395,000 years. So those cycles are almost certainly 406,000 year eccentricity cycles. When we've done that, and we can calibrate using the first appearance of Magnostriatites howardi at 35 million years, and the extinction of Cicatricosis brightes daragensis, then we can put actual dates on every cycle boundary all the way down through here. And then if we map out our packages across the basin, we'll find that some are very widespread, others not, and that we've got some unconformities in here. Those unconformities we can then date very precisely using our age, using the dates applied to our cycles. What this tells us is that in the Coulomb Basin, our Paleogene succession consists of about 5 depositional sequences that are essentially driven by tectonics. And each of those is composed of a succession of maybe 5 to 8 or something like that, depositional cycles that are effectively parasequences driven entirely by glacioclasticity, by eccentricity-driven climate change. And so you can see that it's possible to separate tectonic from sea level change, and provided you've done all your homework. Can this approach be used in Indonesia? Well, the answer is definitely it can. I've made a correlation here to a study that I did with Conoco on North Billet 5, or North Billet Field, about 12 years ago. We saw some depositional cycles there. In the paper, working with a sedimentologist, they tried desperately to think of a tectonic reason for this oscillation, but it's much easier to explain them in terms of glacioclasticity. And so we could identify the same cycles. We find then that the Gabus Formation, which is dominated by widespread lacustrine facies, is a direct lateral equivalent to the Upper Trartan Formation, also characterized by very widespread lacustrine facies, this one here. So it allows you to make direct correlation. I think this approach would work in the Malay Basin, and across the Natuna, across all of the basins of West Natuna. How are we getting on for time? I think we're a little bit late, but let's just work through this as well if you're still all awake. Let's go to East Java, to Java Man locality at Mojokerto, near Mojokerto, a place called Perning. Perning yielded what was thought to be the oldest hominid fossil in 1934, and there was a study about 20 years ago including Professor Zaim from ITB. And we looked again at this succession together with Frank Hoffman, who's a geologist. And the succession there was very accurately profiled by making a profile along the line of this red line just here. And the red line followed the stream bed of the Kali Glagan River just along here, crossed the road, went along the road here into the Perning Quarry. This is the road section, so this is the road along here. Well, anyway, it's the road section. Then we go to the Perning Quarry, which is being quarried for sandstone. These sands are all volcanic sands, and they were deposited in a delta. And you can see the delta progrades. These are the top set beds. These are the progrades, and these progrades have an amplitude of about 30 meters or so, something like that. Then on top of the delta top sediments, we cut deep into the roadside to make sure that we had nice preserved material. And then when we got up to the hominid bed, people were, Professor Zaim was looking here for extra hominid fossils and things like that. And we collected some samples there and analyzed them all for palynology. We took many, many more samples than we actually, we excluded a lot because they were rather unfossiliferous through some of the volcanic sands. But we produced a pollen diagram that looked like this. Now, the yellow here is all grass pollen, and it tells us that our hominids were living in open savanna vegetation area. But if we look at the detail, and I really want to show this with the full screen view, because then I could enlarge this. But if you look at the bottom here, we can see some cycles. One cycle, two cycles, and this whole interval here is a third cycle. Now the reason that's so thick is because it comprises volcanic sands in great abundance. So these were very rapidly deposited, whereas these muds here were very slowly deposited. So the muds form the mollusk 2 and the mollusk, then there's another mollusk bearing interval over here, which the regional stratigraphy is based on. So we were looking at the mollusk 2 interval and the Perning delta succession directly above that. Now, if we look at the mangrove pollen and spores from this, we can find some cyclicity too. Sorry, this is a bit small, but I was hoping to enlarge it, but I can't do that on this screen. So we have a mangrove pollen maximum here, and then the mangroves reduce. A second mangrove pollen, this is Rhizophora mangroves occurring in here, and then the Rhizophora disappears. And then a third Rhizophora mangrove maximum here, and then it disappears again. We can see for this middle section, call it P2, I plotted the mangroves here as a percentage of total mangroves, and it shows that the mangroves in this cycle show a prograding pattern with Rhizophora dominated at the bottom and Nipa dominating the mangroves during the high stand. We can also see the cyclicity in the spores, increased numbers of spores at the bottom in each cycle, just like this. Now, with these data sets, as we have part of the succession in a marine succession and part in a bottom set succession and part in fluvial deposits up at the Java Man outcrop locality, we can use our model that we saw earlier of transportation from delta into marine environment and construct detailed models of the delta as it would have been during the period of mangrove expansion here, which is this model when it would have been basically an estuarine setting. Then with the Nipa dominated prograding mangrove here in cycle 2, which would have been this. And then once the mountains in the south, Arjuna and Willis, erupted, produced lots of volcanic sands, formed this massive sandy delta with hardly any mangroves on it at all, this package here, and that forms cycle 3. So we've got 3 3 packages with 3 separate delta models. Again, using our model of pollen transportation, we can use that to construct the regional vegetation, and we can see that these hominids were living in a setting where we had per-humid montane forests up in the mountains and widespread savanna across the coastal plain. But where does the sequence biostratigraphy bit fit in? Well, it comes in dating this succession. People have already been to this locality, and they produced there have been various attempts at dating this. Swisher used potassium argon dating, came up with an age of 1.81 million years, caused a lot of controversy because it suggested that Homo erectus was older here than it was in Africa at the time. Then Hyodo looked at magnetostrat, came up with a date of about 1 million years, 0.97 thousand years, and thought that the succession was much younger. And then Mike Morwood came along even later, did some fission track dating, got one age of 1.43 and one of 1.49 million years, and these are the error bars, there and there. So can we use the palynology in any way to work out which of these is correct? Well, the answer, we definitely can. We saw from our profile here that we've got 3 cycles, and the cycles are all fairly similar in their magnitude. The later cycle we saw has progrades of 30 meters. That's quite big. So it needs a sea level change of maybe 50 meters or so to generate that sort of amplitude progrades. If we look down here at 1.8 million years, we can't see any patterns that would match this sort of cyclicity. Similarly, if we look up here at 0.97, there's no real patterns here that would match the cyclicity we have here. But if we look at Mike Morwood states at about 1.5 million years, yeah, these oscillations here would exactly be appropriate to explain the sort of oscillations that we're seeing here. Now, we do find in the lower part of our succession, we find Stenoclena milnei spores. And you'll remember earlier that we, working in East Kalimantan, dated this extinction at about 1.5 million years. Now, if we look at the cycles here, there are probably 5 cycles that would be appropriate. But as we've got milnei, we want to pick the older ones. And so my feeling is that of these 5 cycles, the likely ones that are represented here are cycle, the marine isotope stage 51, 49, and 47. Now, if we take the absolute ages of those cycles, which you can read off the Quaternary, standard Quaternary chart, then we can plot this in absolute time, and we have our cycle boundaries at these points, our transgressions at these points, and our hominid bed is dated at about 1.43 million years. That's very similar to the date of the first hominid in Sangiran. So we've used sequence biostratigraphy to date a hominid fossil from East Java. Well, I think we've come to the end. I probably overrun my time, I always do. But some suggestions and conclusions and suggestions. I just like to emphasize that Indonesia offers many opportunities for palynologists to undertake world-class projects. But I'd like to see every biostratigraphy lab build up a collection of modern pollen material, because that's going to make it much easier for you to identify the pollen. I'd like to see more people undertake studies of modern pollen deposition, even if this, as it was for ourselves in Singapore, when we were in Singapore, we did this as a hobby just at weekends. We went up to deposition to areas in Malaysia that we thought would be worthwhile, and we did our studies there. In the Quaternary, we need more sites to be studied that extend to the last glacial maximum, so that we can build a definitive paleoclimate map for the last 20,000 years. We need much more work put into peat swamp studies, because the ecology of these remains very poorly understood, and if we don't understand how these swamps are formed, it's impossible to create a management program for them, or a development program. We need more studies on coastal settings in mangrove swamps. Not only does this provide us with better justification for coastal management and conservation, but all of these act as analogues that we can use in geological interpretation. And each East Java man locality needs careful palynological study using the latest methods, including phytolith and charcoal studies. In fact, Trinel is being studied at the moment in the University of Amsterdam, but there's always room for many more studies on these things. In the Cenozoic, we really need to start publishing a lot of data from outcrops and from well sections. In South Kalimantan, Sulawesi, well sections need to be published. New pollen taxa from the Paleocene and Eocene need to be described and to provide a regional framework of reference sections. Something else, angiosperm pollen from the Tanjung, Nangulan, Malawa. These can be studied using SEM photography of strew mounts, and this could lead to many new identifications and extend the geological ranges of many plant taxa. The accurate identification of Paleogene pollen in terms of extant plant taxa will help clarify the ages of angiosperm clades, and as I said, this is an area of very intense study at the moment among universities. In West Sarawak, it'd be really good to restudy Muller's Cayenne formation, because that work was done in mid-1960s and no one has ever repeated it. The Paleogene pollen record should be viewed in the light of the history of the collision of India and Asia. Equally, we can look at other plate tectonic events and tie those into the record too. And finally, the technique of sequence biostratigraphy needs to be mastered. It's not something that will happen quickly. It's taken me about 25 years to work out how to do it. But the key is for palynologists to work carefully with micropaleontologists. And in doing so, you can build, when you find the same event in foraminifera or an event in foraminifera tying exactly with an event based on palynology, you've got to work out a way in which those two events could have happened. And in doing so, you'll be able to build much more detailed sequence models, including sort of building sequence architecture and landscapes. Sequence biostratigraphy, based on the full integration of palynological, micropaleontological and nanofossil data with seismic, is likely to result in fundamental improvements to our understanding of the geology of Indonesia. Palynology is a key part of the process, but I say it also needs the other disciplines. And palynology important in non-marine and coastal successions, but also in deep marine turbidite successions and deeper marine successions too. So I think we've come to the end of our talk. If you want to quote or use anything from this presentation, you can quote it, I believe, in using this reference. Right, I think that's it. I hope you're all still listening.
Yeah, thank you very much, Bob, for your excellent presentation, for revealing us the powerful of biostratigraphy. You really did very good presentations, and I really enjoyed your presentation. Thank you very much. Now we have two questions. I will read the question because Pak Munasri cannot speak because the microphone doesn't work. And I still welcome for anyone who want to have a question because we still have time, yeah? And Bob will be very happy to answer it, of course. The first one, yeah, the first one is from Munasri, Pak Munasri Aci, formerly from LIPI. Hi Bob, thanks for the presentation. How far the pollen can reach the open ocean? How old is the pollen found? Yeah. That's the
well, you saw from the deep marine core from Lombok Ridge that actually you can find pollen in very deep marine sediments, but of course its concentration is quite low, so it seems to be concentrated in some areas, maybe where currents concentrate it, and less concentrated in other areas. But from the point of view of petroleum exploration, if we have sections that are rich in turbidites or a turbidite bearing, those turbidites will carry pollen directly from terrestrial environments and they'll be rich in pollen. So deep marine turbidites always have lots of pollen in them. And the pollen actually, I think, will tell you surprising things about how the turbidites, where they're derived from, and how the turbidites were formed.
Okay. Yeah.
Yeah, oh, Lombok Ridge. Yeah, Lombok Ridge is 3,600 meters below sea level. So that's, and it yielded quite a lot of pollen. But the guys that did it were very skilled at getting good assemblages out of very poor samples.
Okay. Okay, thank you very much. I think that is from Pak Aci, yeah, from Munasri. The second one is from Sukarno. Yeah.
Okay.
Sukarno. Yeah, Pak Sukarno, you can turn on your audio to ask Pak Bob directly.
Hello, Bob.
Hi.
Hey, how are you?
I'm all right.
Yeah.
So you're still awake?
Yeah,
you haven't fallen asleep yet?
Oh, great. Yeah. I just only one question, Bob.
Okay.
Regarding in the offshore Sarawak. Yeah.
Yeah.
There is in traditional tectonic event, there is the cycle 1 to 8 cycle.
Okay.
Yeah.
The
from late Eocene to Pleistocene. Is it?
Yeah.
Yeah. Did any characteristic of pollen assemblage, mainly from mangrove pollen assemblage across the cycle? I mean, the transition of every cycle. For example, from cycle 4 to cycle 5. Oh, that's all. Bob, thank you.
What do you want? Which cycle do you want first? Cycle 1?
No. I mean, just from cycle 4 and
from
younger.
cycle 4.
Yeah.
Let me just go through all of the cycles.
Yeah, because I'm currently working on the sample from there.
Yeah.
Yeah.
Cycle 1.
Okay.
It's basically the Oligocene, and I think often the Oligocene, probably a lot of the palynology is not processed very well, but when we've had sections that are very carefully processed, they yield good recovery. Very interesting story coming out, and that will be included in the paper we're publishing soon. I don't want to give you the details now, but when that's published, you'll see that. Cycle 2, of course, is a series of prograding deltas, and so that's characterized by your by palynologically intervals that are similar to your that would cover the same interval as your planktonic zones 4, 5, and 6, yeah, and 7 maybe. Cycle 3 is quite nicely characterized palynologically, and that includes the Florschützier semilobata and trilobata events, and you'll be able to characterize it if you've got good recovery with your preorbulinas and things like that. But cycle 4, of course, this is a big issue. Now, the place where we have got cycle 4 studied in detail
cycle 4 is deep marine sediment.
Yeah. But the issue is, what is cycle 4? There's a lot of issue about its definition. And the place where we've studied it in full detail is in the Malay Basin, where we've got a very complete record of what's happening in cycle 4. And so if you compare the Malay Basin with Sarawak, you notice that there are some things missing. And again, this is all coming out in our paper that will be published soon. But so there'll be some stratigraphic intervals missing. I think cycle 4 is very poorly understood. And I still haven't seen a publication that really explains cycle 4 that succinctly, you know, it's because unless you've got a section that has the complete record, it's very difficult to make a judgment as to what is present and what might be missing. And that's the issue with cycle 4. Cycle 5 is, you'll be familiar with that. It's full of late Miocene nanofossils. So does that answer your question?
Okay. Thanks, Bob. And maybe the final one. is your VIM zonation is working in offshore Sarawak as well?
Very well, yeah. Extremely well.
Yeah, yeah, thanks, Bob. Many thanks. Yeah, that's all.
Okay, cheers.
Okay, nice to meet you. Okay.
Thank you, Pak Soekarno, for the question and discussion.
Yeah.
The third one we have from Anupama, India. You can turn on your audio. Anupama, India.
Yeah.
Okay.
Yeah.
Right. So, hello.
Hi.
Yeah.
Yeah. So, I was really thrilled to listen to this because there's so much of information on it. Thank you very much for that.
Okay.
And I was particularly happy that you emphasized the importance of the modern pollen studies.
Yeah.
I think is really important. And then I was wondering about like in Indonesia, and what do you think of quantifying the modern pollen vegetation relationships as it's
absolutely. Yeah. The only problem is with you as a palynologist may not be that skilled at quantifying the vegetation. So meet up with an ecologist and go and study them together. And so the ecologist can perhaps look at the vegetation, and you want to quantify it. You can do that if you're dealing with a forest. You have to use quadrats, and you have to measure the basal area, measure the circumference of all of the trees on the site at breast height. About you measure every tree at about 1 meter above the ground level, and calculate the basal area of that each tree is contributing, and then you compare the pollen with the representation of the trees. Or if you've got a swamp, you use quadrats, and again you work out the percentage of each plant that is contributing to that. But yeah, I mean, probably the best way for a palynologist in Indonesia is to meet up with a plant ecologist. You may already feel that you can do that yourself. Take a transect along a gradient. So the one that I suggested is a gradient up a mountain. Where are you based? You in?
Actually, I'm in India.
Oh, you're in India. Okay.
Yeah,
well,
I'm in India.
Yeah.
And but I mean, because it's tropical palynology, I'm very interested.
Right. Well, there's quite a lot of modern pollen studies that have been undertaken in India. More so than in Indonesia. Indonesia lacks. There's very few modern pollen studies. But meet up with a plant ecologist. Plan out a project where the plant ecologist works out the quantitative contribution of each plant species, then work out which pollen types are coming from which plants, and then you can find out the pollen productivity. And then if you look for differences between the assemblages, you'll be able to work out the pollen transportation patterns. So that's crucial to understanding how, because if we take a sample from a marine environment, all the pollen is transported, and we want to know how to get that into that environment.
Okay. And I mean, that would be great too. I was wondering also if there's any database of for the quaternary palynology of Indonesia.
No, not like. I was only looking the other day at Africa, and Africa have this fantastic quaternary pollen database. So does in South America, but it has never been done for Southeast Asia. So it's something for people to do.
Yeah. Okay. And is there a publication like which compiles the pollen types? You know, the way.
Well, not really. No, the quaternary has always been worked with modern pollen reference collections. But there's a few historical events that have resulted in some disruption of the process that we see in other countries. So, for instance, in Africa, there's always been this strong interest in African palynology by the French. So the French have always made lots of nice pollen keys and modern pollen photographic manuals. In Southeast Asia, the approach was to use modern pollen reference collections, and there were 3 labs that did this. There was ANU in Canberra, Hull which is where I was from in the UK, and Monash in Australia.
Okay.
The only problem was that Hull stopped doing tropical palynology about 30 years ago. It was the government had some reorganization, and they thought, oh, we don't need to do this sort of stuff in a place like Hull, and so it was just came to an end, and so that hadn't continued, and so that's been a major disruption for the process. But there's an active group on Southeast Asia at the moment in Amsterdam, and they're working on Myanmar, and of course yourselves in India, especially at Berbelsani, are interested in looking more regionally. So, does that answer your question?
Okay.
Yeah. Okay. Thank you very much.
Thank you very much, Ms. Anupama, for the question. Yeah. The next question is from Bernhard. Bernhard, you can turn on your audio, Mr. Bernhard. Oh, no microphone. He doesn't have the microphone. I will read it. Let's see. What's the question? I will scroll. Okay, question. When doing modern studies comparing pollen to flora around, how important are seasons? Will these studies give significantly different results in dry or rainy season?
Well, of course. As in England, you know, the flowering is always in one particular season. So the key will be to make sure your pollen sample is likely to be in a position that is going to capture the annual cycle. So you want a locality that will, or a sediment that will not represent just a few weeks of pollen deposition. So some nice wet muds or something like that, or in the forest, it's always moss cushions that are the best for capturing pollen, and they provide. Otherwise, you could use pollen traps. So some people put out special pollen traps that specially trap the pollen, leave it out for 4 or 5 years, and other people have measured the pollen productivity per month. All of these things have been done, but for in your situation, you probably want a long record, and pollen trapping over 2 or 3 years is a good way, but of course that takes a lot of time. But otherwise, use sediment samples. Now, if you're working in in mangrove swamps or in freshwater swamps of any sort, any surface sediment will give you a fairly robust sample. If you're working in, say, in mountains, again, working in the forest, then the environment is fairly wet, and moss polsters, moss cushions will give you the material you want. But in lowland settings, often oxidation will have removed a lot of pollen. So you want to find localities that might have preserved that pollen, and that's what you want to look for. Otherwise, you can use pollen traps.
Yeah, great.
He said,
yeah, I think that's the answer for the question. He said, great, thank you.
Yeah.
Pollen traps, that's it. Okay. Next question from Ashish Mishra. You can turn on your audio.
Hello.
Yeah.
Hello, Bob.
Hi there.
It's audible?
Yeah,
I can hear. Yeah.
Okay. Okay. Good afternoon, Bob. I'm very thankful to attend your lecture, and it was very nice. Actually, I'm working on Dinophyllus cyst in Birbal Sahani Institute of Paleosciences, Lucknow.
Okay. Mm-hmm.
So mainly I'm facing that diachronicity of the specimen, especially like author A suggested the ranges of this specimen is from here to here, and the author B somewhere published in that same specimen with different ages. So how we can remove that kind of diachronicity of that specimen in biostratigraphy to make a certain zones or global zones kind of thing?
So you've got some they're diachronous.
Yeah. Like one specimen is published by different author with different age in a different area.
Okay.
So
again, as I understand the succession of dinoflagellate events. I mean, it's something that probably, again, would tie or would be clarified with a sequence biostratigraphic approach. I know Vandana Prasad has looked at one locality using sequence biostratigraphy, and I thought it looked very successful. So you have to take closely spaced samples from different localities and then look for any pattern of cycles that there might be between the two different areas and see if that resolves the problem. Because with your dinoflagellates, some of them might be restricted to more open marine successions and others are going to occur in shallower water sections. So if you've got a transgression that only reaches a certain water depth, then they may be missing in some of the localities. Okay. So from Vandana's paper, I would say that's a serious, it's seriously worth looking at sequence biostratigraphy to try to resolve that.
Okay.
Oh yeah. Okay. Okay. Thank you, Ashish Mishra, for the question. And then we have the last question. I think this is from Renaldi Faris from ITB. Yeah.
Okay.
Mr. Renaldi, you can turn on your audio. Yeah.
Thank you for your very insightful presentation, Dr. Morley. I'm actually very interested in your sequence biostratigraphic approach because I work in sequence stratigraphy a lot. So okay. I'm just trying to look for insight from you because we know in the Oligocene section in Hack et al. Curve there's a mid Oligocene unconformity, which is like big sea level drop, like 150 until like 200 meters, right? And I just want to know if you see anything special in terms of palynological records in that section.
We've never found it. No. In fact, one of the paper we're producing soon is very much a critique of Hack's sea level curve.
Okay, great. So
okay,
so you didn't find anything special during that mid Oligocene time, yeah? In terms of
I think I know what Hack was seeing. But I don't think it's a big unconformity in the manner and sea level change in the manner that he thinks it is. And the issue is that, sorry, I dry throat. The issue in the Oligocene is that Hack didn't take into account paleoclimate. Now, in the late Oligocene, we have a climate event called the late Oligocene thermal maximum, and it's a period of much warmer climate, and in the tropics that means much wetter climate. And I think what he is actually seeing is the effect of this wet climate interval sitting on top of a drier climate interval underneath that makes it look as if you've got an unconformity. And I don't think his Oligocene sea level cycles make any sense at all. There are sea level cycles in the Oligocene, and my feeling is they're all driven by Antarctic glaciation, and these are summarized in papers by Miller, especially, and co. in the 2008, things like that. If you want
2005, I think,
on if you're working in sequence stratigraphy, it's always really interesting to compare exactly what the sequence biostratigraphy packages are telling you in relation to sequences that you might interpret with seismic, because sometimes they fit together exactly and other times they don't. And it's a really interesting exercise to work out how everything fits together and what the story really is. But to do that, you need really good biostratigraphy. That's the problem. A lot of biostratigraphic data sets are not good enough, and so you come up, the biostratigrapher might come up with some sort of rough questions or rough answers that may not really stand up to full evaluation if you had really good data. So for biostratigraphy, people have got to produce really good data sets with closely spaced sampling in order to get results that are really useful in characterizing sequences.
Yeah, thank you very much for your explanation. I'm actually very glad to hear that because I agree with you.
Okay, we're just throwing out HAC's late Miocene sea level cycles. We don't believe them.
All right. Okay, looking forward to see that.
Okay.
We have one last question from Majida. Yeah, Majida, you can turn on your audio.
Yes, thank you very much. Thank you for the wonderful, really informative presentation. I do have one question about this biozonation, this age diagnostic taxa of this lineage, of this Floroschutzia lineage. I'm wondering whether the biozonation reflects the morphological changes of this Floroschutzia to Cenerazia, because most of the papers are saying that, or is it just in same species? At that time, it was abundant. That time, abundant of this specific pollen type.
It's a definite evolution of of species of Floroschutzia. So again, this is where integrating sequence biostratigraphy and comparing that with the age range, with the ranges of individual species, is really useful, because then you can see, you know, all of these mangroves show abundance changes in each cycle.
Correct.
So if you take any cycle, take any species, if it's in the early Miocene, Floroschutzia levi-poly, it will show lots of the same cyclicity as the depositional cycles, but it won't occur below the deepest, oldest cycle that it occurs in. So, so the answer is, yeah, the two should be separable. You know, so you shouldn't see, you should see the Floroschtzias coming in independent of the oscillations of these species due to sea level oscillations. So definitely due to evolution and not due to sea level change.
Okay, so if you want to date the sediments in the Miocene, would it be feasible to use this palynology? Because in my samples, we don't really have planktonic because we are mostly coastal.
Okay,
so,
you've got some bentonics?
Bentonics, yes, the rotolites, but they are mostly the same in all localities. I'm actually from Brunei.
That doesn't matter.
So
the important thing is, do they show abundance changes?
Abundance changes, yes.
Yeah, well, that's what you need. So you take your foramen. Have you got any nanofossils?
No, unfortunately, no.
Have you analyzed for nanofossils or not?
Yes. We have.
Okay, well, that's. I mean, there are some good examples in Malay Basin that are very similar fluctuations. We want to look for palynological changes which would complement those oscillations. And then we find the age diagnostic palynomorphs that fingerprint some of the sequences, some of the cycles. And then from the stacking pattern, we can name all of those different cycles. Now, if you've got one well, okay, there may be some problems with the first well, but after you've got 2 or 3 wells and you tie them together, cross-check, convert the biostrat to two-way time, compare with seismic and loop tie, then you can see how these all fit together, and hopefully you'll get a nice story come out. Where are you working?
I'm actually in the university, the Brunei University, yes.
Oh, in Brunei, okay.
Yes, in Brunei.
Yeah,
yes.
well, on Brunei Delta, this will work well.
Oh, okay. And the second question is about this Floroschutzia trilobata. I've never seen it in papers because, I mean, I've never seen the picture in papers, mostly at the LM picture, yeah. But most of them, they're actually, like, there are some in Germared, Muller.
Yeah, that's where it's first described.
Yeah. So they have this trilobata, but apparently they look the same. So it's sort of like there are no distinct, they look the same, like as in they took it like copy paste.
Yeah.
Sorry.
Well, maybe that's because you.
So that's why. That's why I'm wondering how does it look like, because I may have it in the sample, but it's sort of That's why I'm still. That's why I'm asking if you have any information on this. I would
Well,
like to know.
I haven't described them myself, but in detail. But I did do some sketches that I put in a book I wrote a long time ago, and maybe that helps. But what I notice is that for Florschutsia trilobata, I see it as quite a wide group of morphologies. When the original paper was written, so much in palynology depends on sort of bending your data to fit with with concepts of species. And this is what happened with Florschutsia trilobata. Muller was quite concerned to describe Florschutsia as not having colpi, because otherwise he would have not been able to use the name Florschutsia.
Yes.
You'd have had to call it Verotricolporites. And he didn't want to do that because he wanted to honor his research supervisor when he was a student, who was his name was Florschutz. And so he was desperate to find an important species, an important genus that he could name after Florschutz. And so he limited the delimitation of Florschutsia to exclude colporate grains. But then when I started looking at these, so there were as many colporate ones, and there were lots that were not trilobate, but they all fall within the same grouping.
Exactly.
So,
it's like Lagostromia.
Well, Lagostromia differs because it's got well-developed polar colpi, and that's quite distinctive.
Okay.
And these have been Lagostromia has been published. Nice images, again by Muller, who tried to sort this out in the 1970s. But understanding this whole complex, it needs someone, perhaps like you, to photograph and document all of these, because there's a lot more morphological variety there. I mean, I've tried to do these things, but I've never had time to describe all of the different, to photograph all the Florschutsia trilobata varieties. But basically, with Florschutsia trilobata, you can have forms that are trilobate or that are not trilobate. You can have forms that are colpate or colporate. Sorry, you can have forms that are porate or colporate, and you can have forms that show really obvious exene structure and ones without exene structure, and that gives you 8 different morphotypes within Florschutsia trilobata group, and that excludes your Lagostromia. It's a really interesting group. It'd be worth spending a lot of time on if you've got time and in a university environment to photograph lots of different types. And I think if you need any help, then I'd be interested to know what you were getting.
Okay. Okay. Thank you very much for the question. I think that will end our discussion, Bob.
Okay.
On behalf of FOSI, I would like to extend our honor, because we are very honored to have you in this. Unfortunately, we can meet not face to face.
Sorry?
So next time you come to Indonesia, FOSI will be very happy to conduct a meeting to have a sharing with you.
Yeah, well, we'll see when we can. We'll try to get to. We'll try and get to Indonesia soon.
Yeah. And then we will upload this talk to YouTube later, so everyone who cannot.
Yeah. So we're not keeping away from Indonesia because we don't like you anymore.
Thank you, Robert.
Okay.
We'll be there when things ease up and we can start traveling again.
Yeah, thank you very much. Yeah, I think that's from me. And then Melinda, do you have something to add?
Maybe from me, not much. Yeah, indeed, this one is a powerful presentation, Bob. So I really enjoy your presentation, and please let us know whenever you come to Indonesia, and Santi as well. Hope to meet you in person after the condition back to conducive. So
you might be able to you won't recognize us because we'll have a mask on.
Yeah, much yes. So keep in touch, Bob. So maybe lunchtime already in UK. So enjoy your lunch. And
yeah,
Santi,
nearly lunchtime.
Yeah, many thanks once again. And Next week, we will have a talk from Peter Lan on the stratigraphy in Southeast Asia. So maybe after we learn more, learn a lot about palynology, and then we will talk about stratigraphy.